In thalas, in generally, they are grows in aquatic and terrestrial. That is common and also shows symbiotic nature also. What is symbiotic nature? Some of the algal synophytes member shows the symbiotic nature. What is symbiotic nature? What is meaning of symbiotic? Symbiotic meaning. Don't you know about that?
Symbiotic means
Symbiosis.
they are helpful for both, like in
Right?
environment also.
In symbiotic relationship, both are get mutual
Both
benefit.
get, yes,
Both get mutual benefit. That is called symbiotic generation.
symbiotic.
So, however, a habitat actually it is not an important one. Habit about right about habitat never ask in examination. But
Okay.
anyway, in part of algal general, sorry, Sinophyte general character, we should mention about habitat as a first heading.
Okay.
So
Okay,
in
sir.
general, we have to write about in habitat, aquatic common, apart from the aquatic, terrestrial also,
Terrestrial.
right? And
Yes.
also show symbiotic nature also. So
Okay.
these are the commonly appeared. This type of habitat commonly we can see in general. And where they are, just wait. And after the distribution, now what we discuss, one is completed. Cyanobacteria can be found in almost every terrestrial and aquatic habitat given. Apart from that, you can mention in our page number 69, 6.3 general character, and also they can grow as a symbiotic also, symbiotic nature also. Symbiotic, just add that word also, symbiosis, symbiotic nature.
Yes.
So that much enough the first point given, general character first point about habitat, that is clear. And what is given in second character?
Include unicellular
Yeah,
and
Right.
unicellular.
That means second heading should be thallus structure or thallus diversity. Thallus diversity or organization of thallus. Put heading there, thallus organization. Second heading, thallus
yes.
organization. Write down the heading, thallus organization. Right. Let's
Mm.
see. Let's see the thallus organization. Where it is. Just wait.
Sir, while writing the answer, we need to mention these headings and we have to write these points, sir.
Heading. You can mention the heading.
Okay, sir. Thank you, sir.
Where is? Are you able to see the share slides?
Yes, sir.
No, sir, it got stuck at objectives.
Okay. Just wait. Right. Now, are you able to see? Hello? Are you able to see the general character?
No, sir, it is coming. Files are coming.
No, sir.
Now clear?
No.
See the Now, are you able to see? Right?
Yes, sir. General character.
Right. So in general character, first actually, don't forget here the heading is habitat. The most widely distributed any organism, algae, and any habitat, marine and freshwater environment. Of course, in aquatic, marine water and freshwater environment. And also, terrestrial means moist soil and also rocks are
On
either in
land.
the right and free living and also here given symbiotic also. Here, write down. Just wait. Let's change. Take the pen here. Freshwater, moist soil, rocks, free living and symbiotic nature, symbiotic organisms also. Right? Some planktons. Planktons mean they are the free living inside the water. And after that, after first heading, second heading, this is the second structural range already given in your, but actually not clear in our book, but add this one. In our book, only given 1 point, but don't write 1 point. Just you can elaborate. You will have to elaborate like this. The structural range from unicell, unicell through branched, unbranched filamentous, branched, unbranched filamentous, and also unspecialized colonial aggregation, colonial but not clear, like a volvox. So anyway, unicell present in this Cyanophyce bacteria and unbranched filamentous present and branched filamentous present and also not given and pseudo branched also. You can add another word pseudo branched. Pseudo branched also present in Cyanophyce thallus. Pseudo branched. Just add this one. Sorry. Pseudo branched. Right?
Yes, sir.
So I will show you the diagrammatically later. Unicell organisms over the filamentous organism, like that. And so these organisms dominated in ocean ecosystem rather than the freshwater. Mostly the Cyanophyce members dominate in ocean marine water ecosystem, right? And we were given clearly in this slide. Just wait. Right. Where is pen? Habit or thallus organization. Again given clearly here. Just see. Where it is. Where is pen, man? So anyway, in habit we can see unicell. Already told you first unicell to multicell. Unicell they are singly the best example. Are you follow my cursor?
Yes, sir.
Syncostistis best example. On colonies of unicell, aphantosi. And next one, these are filamentous. And first unicellular uh, syncostistis and aphantosi. These are the for unicell. In unicell also one singly and also colonial. Colonial unicell, one example, and single, one example. Right? Let's draw this diagram. Actually, unable to take the pen here. So anyway, I will show the diagrams later. Right. Now next, if you see the row of cells unbranched filamentous without sheath, example oscillatoria, and also in our syllabus given NASDAQ already you must have learnt it during practical training classes about the thallus structure. Here the thallus structure of the oscillatoria and NASDAQ both are belongs to Synophyce members. The thallus is unbranched. There is no branches and filamentous without sheath. Without sheath called as trichome. Without sheath called as trichome. With sheath actually called as filament. Best example oscillatoria, and also you can write the NASDAQ also. And many trichomes in large sheath. Many trichomes. That mean in oscillatoria singular, it's not a group. But if many trichomes as a group form, that is called microcoelos. And next one, unbranched filamentous with sheath, lingbaya. Without sheath trichomes, actually they are called trichomes, and unbranched filaments with sheath. First one without sheath, and second one with sheath. That means actually what happened here? I'm unable to take my. What happened to pen? Right. So what happened in first type in oscillatoria in oscillatoria actually what happened here? The thallus unbranched filamentous. This is filament. And these are the cells. Without sheath. There is no sheath. So this is called trichome. But what in Lingbaya, if you see in Lingbaya, same unbranched thallus, like the cells appear like this. These are the cells. Of course, maybe the cells does globose structure and rectangular structure, whatever it may be. So this is also unbranched. But here, let me change color. In Lingbaya, what happened? It is covered by sheath, blue color, covered by sheath, covered by sheath. Lingbaya. And these are all uniseriate. What that means? One cells arranged above, uniseriate. And again, the uniseriate branched. The next one, the branched filamentous. Take branched filamentous. Masticocladus. The branched means simple. For example, this is the Masticocladus. What happened here? Branches are there like this.
Okay.
Simple, we can. There are also some branches like this. So this is the branched filament. But uniseriate, both are uniseriate. And if you take multiseriate branched filamentous stigonyma, and just now I told you, and just add another word that is pseudo branched filamentous cytonyma. So anyway, let's see the multiseriate. Multiseriate means cells are not uniseriate like this cell, above the cell, above the cell, above the cell, and also side the another group of cells, 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 maybe 2 to 3 rows like this, and branched, and with branches. So what happened in first diagram? Only one uniseriate, one line. But here, how many series? One, and
Multi-series.
right, two. So it's, and also maybe more than two also may be there. So this type of condition called as multiseriate steganoma. And another one is called pseudo branched. Pseudo means, what is meaning of pseudo? What is meaning of pseudo?
Pseudo means movement, sir.
Pseudo means false, not true. Pseudo means false, not
Yeah.
true.
Yes.
Pseudo means false, not true. Uh, Best example. That means, let me draw diagram. Actually, it look like a branch, but not branched. There is no place, and here I will draw. This is one branch, filamentous, and this is another filamentous. Both are different. Both are different cytonema. Here, how many cytonema? Two cytonema. But the two cytonema attached with slime layer. Attached by slime layer with this attachment, it look like a branched one. It look like a single one. It is a single with branched. But they can easily separated. These both are each other easily separated. And that's why it is not a true branched. It is appear like a branched, but not true. This type
Yes.
of this type of thallus called as pseudo branched. Example
Cytonema.
cytonema. Don't forget the headings and also diagram. Just write the rough diagram. That is enough to you, right? So update completed and thallus structure also completed. What next? First of all, let's see the diagram. If you have any diagram, we will check. After thallus diagram, after thallus structure, what is next actually? In heading, what we learn?
Reproduction.
What about cell?
Cells, yes, sir, cells. Cells and then reproduction.
Then after that, reproduction.
Reproduction.
Let's see. So pseudofilamentous completed one thallus. And thallus organization, already told you, thallus organization means you have to write from the unicell to multicell, right? The same, non-filamentous, filamentous. It's the same again, repeated, given. And cell structure. Next, we will discuss about cell structure. As we said earlier, the cell actually, in here also, there we should follow order. Order is actually here what they given, the first of all the cell. What is the components of the cell? Already we discussed in general character. Already we discussed in general character.
Yes.
Tell me
Chloroplast.
in order. I'm asking about order.
Order. Nucleus will be there, sir.
Nucleus,
Cell wall.
right? So first cell wall.
Cell
First
wall.
cell wall. Next,
Chloroplast maybe inside. Cell
actually,
membrane comes, sir.
cell membrane also present.
Cell
Cell wall,
membrane.
cell wall,
Cell organelles.
cell organelles.
Nucleus.
Just wait. This is cell wall. And cell wall maybe in some of the cyanobacteria, it is covered by capsule also present like this. This is the capsule, this thick strip capsule, cell wall and capsule. After the plasma membrane, this inner layer called as the plasma membrane. And as you said, the cell organelles. What type of cell organelle present in the cyanobacteria? What type of cell organelle present? Is it eukaryote or prokaryote? First of all,
Eukaryote.
right? As it eukaryote, tell me the type of cell organelles. Tell me type of cell organelles. Is it eukaryote or prokaryote?
Sir, eukaryotic.
Cyanobacteria.
The prokaryotic.
Cyanobacteria, bacteria, eukaryote or prokaryote?
Prokaryotic bacteria.
Right, tell me what are cell organelles in prokaryote bacteria? What type of diagrams? What type of cell organelles are you able to see in diagram? And also in slide.
Thylakoids, ribosomes, DNA, gas vesicles,
That's it, that
chlorophyll.
much. Only remember, actually, in general, in eukaryote, what type of cell organelle present in eukaryotes? Mitochondria, Golgi complex, lysosome,
Yes, sir.
endoplasmic reticulum, these are all present. But in prokaryote, only 70s ribosome present, only 70s ribosome.
Yes, sir.
Okay, sir.
And here thylakoids also not fully developed. As it prokaryote, chloroplast not full developed. Instead of chloroplast, one part of the major part of the chloroplast, thylakoids freely appeared. Actually, the thylakoid present inside the inside the chloroplast membrane, chloroplast layer, but there is no chloroplast layer here. Only the free thylakoids are present. We can see in slide free thylakoids and 70s ribosomes and gas vacuole polyhedral bodies and gas vesicles. These are present. And apart from that, you should write in order first cell wall and maybe the cell wall covered by capsule. Most of the BGA covered by capsule also and plasma membrane. After that, cell organelles. In cell organelles and actually before cell organelles, you should write about nucleus. Not given in here also. Tell me about the nucleus in cyanobacteria. Nucleus as it prokaryote. How the nucleus?
Nucleus.
Nucleus actually without protection, nuclear membrane, nucleolus.
Yes, sir. No membrane.
No membrane, only the threads are present. That is DNA. Like this, we can see in diagram also. Only the threads, the DNA like threads are present without protection of membrane. Right? That is called genetic material. Free genetic
Naked
material.
DNA.
Right. That is called naked. Naked means without protection. So naked DNA present. And we can observe here thylakoids. Here they are the thylakoids. The thylakoids are freely present in cell. So these are thylakoids. And gas vascules also present. Polyhedral bodies, gas vesicles, they are all present inside the cell. And already in our book given what is the chemical, cell wall chemical. Peptoglycan mucopeptide. Actually, the plant cell, what is the major chemical composition? Plant cell in general.
What?
Hello? A plant cell chemical composition. I'm asking in general, not about cyanobacteria.
Chlorophyll.
I'm asking about the cell wall chemical, not chloroplast.
Peptidoglycan.
But cyanobacteria peptoglycan. In general, plant cell cellulose. Remember it. The plant cell chemical composition cellulose. But in cyanobacteria, peptoglycan, mucopeptides, they are the present in cell wall composition. Right? Is it clear about the cell structure? Hello?
Yes.
Right. After cell structure, now we can go through the reproduction. Now tell me the reproduction. How many types of reproduction? Already you must have learnt in general character of algae. Tell me the type
Vegetative,
of reproductions. Right. Three types. They are vegetative,
sexual
vegetative,
reproduction, asexual
sexual
reproduction,
reproduction, asexual reproduction, and
only three are those.
sexual, sorry, vegetative, asexual, and sexual reproduction. Three.
Yes.
Three are present. And in cellular organization, already in free thylakoids, we just discussed about free thylakoids. In free thylakoids, just take pen. In thylakoids, pigments are present. What are the pigments? Are chlorophyll A and blue and red
Yes.
phycobilins. Phycobilins, they are called phycoerythrin and phycocyanin, allophycocyanin. So these are phycobilins, blue and red phycobilins. And apart from that, carotenoids and xanthophylls also present inside the cell. So in order for cell wall, capsule, plasma membrane and nucleus, cell organelles. In cell organelles, we will discuss about the thylakoids. Inside the thylakoids, pigments are present. What are the pigments? Chlorophyll A and phycobilins and carotenoids and xanthophylls. These are the necessary pigments inside the synophysis member. Right? This is all about the cell structure. Next, after cell structure, now is there any diagram? Let's see. And another thing is inside the wait, this shows the photosynthesis. Does cyanobacteria show photosynthesis?
Yes, sir.
As it
Yes,
as I
sir.
told you,
As it has chlorophyll, it shows photosynthesis.
right, as it presence of chlorophyll A, it shows photosynthesis. After photosynthesis, reserve food material prepared. Here the reserve food material is synophysian starch. In general, starch is present, but in synophysis member, synophysian starch is present. Right? So this is the reserve food material in synophysis member. And remember the photosynthesis takes place in free thylakoids with help of the chlorophyll pigments. Only chlorophyll A present and also phycoerythrin, phycocyanin also present. Here the phycocyanin, phycoerythrin importance, which gives the pigmentation color to the body. If the phycocyanin rich, they are in green in color. If the erythrocyanin rich, they are in red color. So anyway, the synophysis members appear in blue-green color or in red color. And sometimes it appear as a red tides, red sea, red snow because of the group of synophysis members as it composition of rich amount of erythrocyanin. It is completely that locality completely appear as a red color, right? Next one. So these are the headings. These are the thallus organization diagrams. Just now we will discuss. Just now we have discussed already. Unicell, filamentous, spirulina. This is also filamentous unbranched. So here the given different type of thallus also. These thallus diagrams present in our book also. Right. Classification no need. Not much important about the classification of the synophycy. Now immediately go through. Reproduction. Now vegetative reproduction. Vegetative reproduction. Vegetative reproduction mainly takes place by fragmentation. Actually not given here. Fragmentation or by cell division or hormones, pseudo hormones. This much. And endospore, exospore, that is by asexual reproduction. Right here, what happened? Just wait. Actually, in our slide, asexual reproduction, binary fission. Actually, the binary fission comes under the vegetative reproduction. But whatever it may be, we can write in both category. Actually, the most appropriate, it should be in vegetative reproduction because here the entire cell, entire vegetative cell divided into two. After divide into two cell and these two individual cell can grow as a individual thallus. That is called binary fission. So now we can see here the vegetative cell. Now the vegetative cells elongated and form a constriction. This constrictions elongated each other and as a result centrally form as a septa. After formation septa, along with the septa, these are divided into two. And these two individuals developed into two thallus. So this is happens in mostly in single-celled single-celled thallus of the cyanobacteria. And next one is fragmentation. Either given, actually fragmentation not given, but remember fragmentation is the very most common method in filamentous thallus. Let's see how the fragmentation happen. For example, this is the fragment thallus, that means oscillatoria thallus, filamentous unbranched, right? Filamentous unbranched. This filamentous unbranched. Filamentous unbranched. Break into pieces. Due to any reason, aquatic animals or water tides break into pieces. For example, it break into three pieces. One, here the two, and last one, three. Now later, this each and every fragment can develop into new thallus, new oscillatoria. One that like that, how many oscillatoria are formed after fragmentation of this method? How many oscillatoria are formed after fragmentation? Three are formed. That's it. So this is called fragmentation. And the next one, hormogony also look like a fragmentation. Hormogony also look like fragmentation, but the most important, they are not mechanically separated. Some with help of the weak cells among the filamentous thallus. Where it is? For example, this is a filamentous thallus. Among the filamentous thallus, some of the weak cells are present. For example, this is the weak cell. Due to the weak cell, can easily separated into two parts. This type of method called as hormogonia. Remember, hormogonia look like a fragmentation. But in fragmentation, there is no weak cell or all healthy cell. But here break into by aquatic animals or water tides. That's it. But in hormogonia, necridia type of weak cells are there. Here from the exactly necridia cells, as it is weak cells, it is completely separated into parts. For example, it is divided into, for example, inside the thallus, three weak cells are present. It is break into three pieces. That's it. So this is this type called as fragmentation method. And another one is endospore formation. That is now next one is asexual reproduction. Asexual reproduction, by spore formation. Already told you in algae general character. The spores are developed inside sporangia. Let's take next this way. And also we should learn about the heterocyst also later. But before heterocyst, movement of we will list as the ecological importance. Cyanobacteria production, cyanotoxins. Okay. Actually, are not given, but where they given some sanatorium. Right here. So this is remember always asexual reproduction takes place by means of hello. Asexual reproduction always takes place by means of spores. Maybe they are the endospores or maybe the exospores. Endospores means the spores inside the for example, this is the thallus, this is the thallus, these are thallus cells, thallus cells, these are the thallus cells. So in this thallus, one of the cell act as a sporangium. Later the entire cell protoplasm rounds up and here, take here, rounds up and developed into spore. This is called endospore because it is formed inside. If the same spore formed outside the thallus, outside the cell, this is called exospore. Exospore. And the same nanocytes, echinates formation also formed like this. We will discuss later. So anyway, whether it is endospore inside or exospore outside, after formation, after maturation, these are released out. And these spore, when the favorable condition or in appropriate condition, optimum temperature, optimum moisture, can grow as a new thallus. Can grow as a new thallus. Can grow as a new thallus. Can grow as a new thallus, right? So echinate formation during unfavorable condition. What happened? For example, this is the thallus. This thallus, what happened? Automatically, any one of the cell developed into thick cells, synthesized into thick cells. Synthesized into thick cells. After formation, thick cells. This is called echinate. Already told you it is formed during uh, uh, unfavorable condition. In unfavorable condition, all are disappear except the echinate. These echinates survive, and when the condition reoccurred, optimum condition reoccurred, again germinate into new thallus. So this is all about asexual reproduction. And if you see the sexual reproduction, actually sexual reproduction absent in cyanobacteria. Remember it. Sexual reproduction absent in cyanobacteria. Okay, is it clear? Right. Now, one thing we'll discuss, that is movement of cyanobacteria and heterocyst. Let's discuss about that. Sorry. Movement of heterocyst. Actually, heterocyst is the specialized type of cell. Where it is? Here. Heterocyst, actually the specialized type of vegetative cell when compared with vegetative cell, which is enlarged in size, thick, and it is actually photosynthetically inactive. Photosynthetically inactive means already we discussed in most of in vegetative cell components. But if you see the heterocyst cell, in heterocyst cell, the very less thylakoids are there. Less thylakoids means less pigments also present. Without pigments, can it perform the photosynthesis?
No.
No, that's why it is inactive. And another importance of heterocyst, it is utilized for nitrogen fixation. It is utilized for nitrogen fixation, transfer of metabolic and ammonium. Instead of that, just write one word, the heterocyst function, nitrogen fixation, and also this heterocyst involved in reproduction also. Heterocyst involved in reproduction, vegetative reproduction also. Right? This is the all about the heterocyst. And next, let's see the movement of the thallus. Here, the movement of thallus also not important in exam point of view. In exam point of view, only remember only one, that is general character. In general character, no need to write all. Just in general character, first distribution, habitat. After that, thallus structure, cell structure, reproduction. This much enough.
Yes.
So there are photosynthetic inactive, already told you, because they loss of photosystem 2. Thus, especially in chloroplast, photosystem 2. Actually, there are two types, photosystem 1 and 2. But in heterocyst, lack of photosystem 2 system do not fix carbon dioxide, and not do they produce the oxygen. They also exhibit high rate of respiration and oxygen consumptions. Already told you the heterocysts are formed regular interval, that means intermixed with vegetative cell. Maybe it will deposit situated in middle of the cell or middle of the thallus or top of the thallus. Movement of the cyanobacteria, gliding movement, swimming. Gliding movement means slippery movement. Gliding movement, active movement of an organism in contact with solid substrate. What happened during in solid substrate, with help of the mucilage sheet, it shows the slippery nature. So the slippery nature called as gliding movement. So mechanism already told you just now, mucilage layers microfield generates the waves. Next, swimming, the active movement in organism without contact the substrate. In gliding movement with the solid substrate. But in swimming without contact substrate, we can swim in water. That's it. And already told you symbiotic association with lichens and symbiotic association with azolla. Lichens, what is lichens? Lichen symbiosis, both algae and fungi.
And fungi.
What?
Algae and fungi, sir. Like
Right, algae and fungi. Here the algae group is mainly cyanophyceae and chlorophyceae members. And in fungi, mainly Basidiomycotina and Ascomycotina fungal. Fungal members form the association with the algae. And Azolla water fan also shows symbiosis nature in Anabina. And liverworts, bryophyte also shows symbiosis nature with the cyanophyceae members. And coralloid roots actually not given in gymnosperms. Symbiotic nature exhibit in the form of coralloid roots. And ecological importance, the blue-greens algae are primary colonizers are bare soil and rocks. That means the composition, due to the habitat of the rocks, actually in the beginning we had discussed they are aquatic and they can grow on rock. What happens if they are in rock? Actually, can you find out any vegetation on rocks? Can you find out any vegetation? On rock, can you find out vegetation? Actually, there is no vegetation on rock in general. But what happened? Lichens are some of the cyanophytes members in the form of lichen. They can grow on rocks. As it grow on rocks, weathering form. Due to the weathering, the soil formation surround the soil. Due to the soil formation, now this is available to the development of the plant. It gives the proper habitat to the plant development. And ecologically, another important, already told you, with help of heterocyst, it fixes the nitrogen. And the blue-greens contribute the water plankton's food chain. What is the function of water plankton in food chain? How it is functioning ecologically? What happens if there is no water planktons? Hello?
for aquatic animals.
Right, it is feed for aquatic animal. Aquatic animal feed for another aquatic animals. So this is the cycle.
Food
So
chain.
for the biodiversity balancing, we should all compulsory deposited in water ecosystem, right? Without water plankton, there is no zooplankton. No zooplankton, there is no fish, no big fish. There is no ecosystem. There is no biodiversity. That is the ecologically importance. And toxins. They form the toxins. As the formation of blooms, already we must have you must have seen in our surroundings some of the polluted areas. In polluted areas, like a scum layer, we can find out some of the rivers, some of the water ponds. These scum layers called as blooms. The blooms mainly developed in developed in not a fresh water. They are polluted water, mainly appeared in polluted waters, and it is cause for mostly gastrointestinal diseases in cattle and humans due to the toxin nature. These are produced some cyanotoxins. The cyanotoxins are neurotoxins, alkaloids that target the nervous system. Neuro means nervous. The already given the example, anatoxin and saxitoxin. What are the symptoms? If we take water, if we drink this type of water, planktonic water, sorry, this bloom, organic bloom toxin water, they are staggering, muscle twitching, gasping, this type of, and also headache, stomachache, and gidding nature, they are all appeared. And hepatotoxin, neuro means it effect on the neurological mind, nervous system. Hepatology means study of liver. Hepatoxins, the effect on the liver, right? Remember, the effect on the liver here. The hepatoxin example, microcysteines, nodularians. Symptoms, weakness and vomiting and diarrhea. These are the symptoms of neuro. And here the given table. This table no need. And also let's see the some of the difference between bacteria and cyanobacteria. The bacteria cell comparatively small, BGA cyanophysi long, large. In bacteria flagella present, but cyanophysi no flagella. Both are heterotrophic. Sir, here the bacteria shows heterotrophic. But cyanophysi only autotrophic. And fourth one, they are anaxenic photosynthetic. They are axenic photosynthetic. Anaxenic means without oxygen photosynthesis. They are with presence of oxygen photosynthesis. Aerobic, anaerobic, but they are always aerobic. Reserve food material glycogen. Already we discussed reserve food material is cyanophysin starch. Is it okay? Any doubt in cyanophysi? Hello?
No, sir.
Right. So now tomorrow class we will discuss about fungal general character. Fungal general character. If you learn fungal general character, you can easily, and also fungal general character, some of the fungal disease also. These are the most important to an exam point of view. Okay? Right.
Yes.
Thank you, one and all.
Thank you, sir.
Thank you, sir.